when ecologists are studying populations of animals or plants, they often need to measure how big that population is, and then they can study whether it's changing over time. So there's a few different ways that scientists have come up with of measuring the size of a population. And it really depends on the type of organism that they're looking at. So the easiest method is just simple counting. The next method we'll look at is called Quadrats, or quadrat sampling. Another method is called Transect sampling. And the final method is called Capture and Recapture. Capture, recapture. So counting, it can be used for lots of populations, lots of different types of species, but it's not very accurate. The next two, quadrats and transects, they're really good for species that don't move at all, or that move really slowly. So species that are slow. So plants are really good for quadrats and transects, or marine animals too, like limpets that you find on rocks, or sea stars that you can find in rock pools. So it's easy to sample them and they don't run away when you're trying to count them. Capture and recapture, that's what scientists use for most animals that are difficult to catch or that move around a lot. So let's look at quadrat sampling first. So this scientist here at the rock pools, they're using quadrat sampling. And the method for quadrats is scientists will take a map of the area and they'll draw a grid on top of it. So just like the grid you can see on this page, they'll look at a map of the forest they're looking at. So there might be rivers and forests, and then they'll pick random grids in that area. So they'll use a random number creator, and then those grids, they'll go and take a quadrat, which is this, it's really just a square made of wood or plastic. So they take their quadrat tool and they place it down on the ground and they count how many species are in that area. So they choose random grids, then they measure out those grids and place down their quadrats, and then they count how many limpets are there in that little box. They count the individuals. And then there's some maths that scientists have come up with, and they can use maths to estimate how many individuals there are in the whole area that they're looking at. So number 4 is they use maths to calculate the total. and scientists can be pretty accurate with quadrats. The next method is called transect sampling. And transects, they're really good when you want to look at how a population changes across an area. So we can see in this diagram, if we want to look at how the population changes from right down at the ocean, all the way over the dunes, down behind the dunes, and then over into the fields behind, we could use a transect to compare. So transects are a line drawn along an area. So the method scientists use for a transect is, firstly, they'll draw a line and measure it out with a tape measure or along a map. Draw a line along the environment. And then they move along that line or transect and they count how many species there are along that line. They count the species, or they might just be looking at one species, so they'll count the individuals. And then usually for a transect, they'll draw a diagram or a table that shows all their data that they've collected. And that helps them to analyse which species prefer to live where. The next method is called Capture-recapture. So this is the one that's good for animals mostly that can move around. Capture-recapture. So this method means that scientists actually have to catch the animals, and they use the most friendly methods they can. Often it's really big nets that don't tangle up the animals, or it's cages, but they release the animals as soon as they can, so the animals don't get too traumatized. And then they put a tag of some sort on the animals. So the first step was catch a group or a sample of the animals. The next step is mark them or tag them. So you can see these swans here in Melbourne, they've got a tag on, and scientists have captured and marked nearly all the swans in Melbourne by now. And they put these little rings around their neck, which don't harm them at all. They've put a lot of work in to make sure the animals aren't harmed. And numbering them and tagging them helps the scientists to study the swans and see where they go. So once the animals are tagged, then they release them back out into the wild, and they wait for a while. So that means all the animals that are tagged, they'll eventually mix in with all the rest of the animals in the population again. And then after a while, they recapture a new group of animals out of all that randomized population that's been mixed up. So you can see down on the diagram here, once all the tagged animals have been released, they're all mixed together with the non-tagged animals. So when you recapture a group of animals, like they're doing with this fish here, then some of the animals there are tagged and some of them aren't. And there's some pretty simple maths that scientists have invented, and they can use it to calculate how many animals there are in the whole population. So calculate the total population. And this method is really accurate for counting the number of animals in populations.